Thank you very much, Umesh. Michelle, if you're here, for the invitation. The organization, thank you very much for organizing the flights and the hotel.
You're welcome.
It's wonderful being part of this program, and I'm grateful to David for including me, and to Agnes for all the hard work and the molecular analyses, and to Robin for doing a lot of the in vivo work. So Keiichi, who's here, for working with me in Boston on this program. So I've got no financial relationships, alas, relating to this project. And David asked me to speak a little bit of overview about the vascular biology of transplantation, and why it is relevant that we need some of these new synthetic heparan sulfates to deal with some of the rejection crises, ischemia-reperfusion, and other vascular forms of injury. Essentially, vascular biology transplantation revolves around the endothelium. And if you look at the total number of cells that we have in our body, in the endothelium, it's of the order of 6 × 10⁻13 cells, weight around about 1 kilo. So if you can imagine about 8 Big Mac patties, that's the sort of your endothelium. And if you imagine spreading that around 2 or 3 American football fields, that's basically the layer that you've got. And essentially, whether you're looking at artery, vein, capillary, you're looking at a monolayer of cells, a very thin blue line standing between your body and the circulating blood. And the microvasculature itself is also unique. There are unique properties whether you're in the liver, which is allowing you to take in chylomicrons, very large particles, part of the digestion process, or in the brain, where there's absolutely no leak, there's a very firm blood-brain barrier. So there are unique spatial and temporal features of the endothelium in the microvasculature, and this dictates multiple functions, whether you're allowing lymphocytes to get into a lymph node, whether you're preventing lymphocytes from entering the brain, for example. This is all dictated by the endothelium, and then it's also these functions are associated with pericytes around the vasculature, which are also important in regulating the permeability and the functions of the microvasculature. So in transplantation, again, when you do a transplant, you are removing an organ. It's subject to a period of ischemia, lack of blood, and then you have to reimplant it. And that leads to a process of reperfusion. You're then subjecting that graft to antibodies, complement, cells, and then you're getting rejection, either acute rejection, which occurs very suddenly, or the chronic rejection, which occurs over months to weeks. And this is the same whether you're actually looking at liver or kidney, or whatever graft you're looking at. So I'll give you these examples of the microvasculature and the blood vessels and how they respond to transplantation stress, using the liver as an example. So the normal liver situation, you have the sinusoidal endothelial cells, you have Kupffer cells, you have NK cells and so forth in the actual microsinusoids of the liver. With ischemia, you end up with endothelial activation, macrophage activation, and you end up with neutrophils, fibrin, and red cell sequestration within the sinusoids. With reperfusion, you lose the heparan sulfates, which are very low level in the liver, but they're still there, and you end up with this apoptosis, necrosis, platelet adhesion, and activation in the microvasculature. So you can keep a liver graft, for example, for about 12 to 15 hours on cold ischemia with preservation fluid, lungs of the order of several hours, and as David, who is a cardiac surgeon, will tell you, hearts really should not be transplanted beyond 4 to 6 hours in preservation. Kidneys you can put into a bucket, keep them for a day or two, well, a day, 24 hours. You can fly them across the country. You really can't do that with lungs and hearts. So the graft obligatory has to have a period of ischemia, warm ischemia when you're taking it out, cold ischemia, then warm reperfusion type injury. The graft survives that. Then you subject to allorejection, which affects the vasculature. Now, in the liver, you can have a cellular type rejection, which is T-cell mediated. But this, again, is associated with endothelial cell damage. One of the cardinal features of rejection is that you're damaging the endothelium. So in the context of just an immune response, the target is the vasculature. Many of the manifestations of that rejection are associated with destruction of the microvasculature and fibrin deposition, platelet sequestration. Platelets are much more important with humoral rejection, which occurs in an allograft. Very unusual to see it in the liver. You do see it with the heart, you do see it in the kidney, and that's associated with fibrin deposition and lots of platelet sequestrating. And that's what we see when we also transplanting organs across species. We see a lot of humoral rejection with these manifestations. Now this is chronic rejection, months to years. What's associated here is that you get progressive near-intimal hyperplasia. So the lining of the endothelium is replaced by a pseudo-intima, neo-intima, proliferating cells which eventually occlude the arterial lumen, and you lead them with progressive organ ischemia. Downstream of this, you will see neovascularization, angiogenesis, such that you would see with maybe diabetic retinopathy, in tumors, for example, with new blood vessel formation, secondary to that ischemia. So let's talk about the microvascular and the heterogeneity. Clearly, if you're transplanting a liver or a kidney, there are different vascular responses. The microvasculature also in the lung is unique. It is obviously subject to environmental stresses, the inhaled gases and so forth. And the tumor microvasculature, again, is very leaky, very defective, high rates of proliferation. So all of these are quite different in their own way. And again, I alluded to this earlier, with preservation injury, lungs subject to very, very acute injury, subject to ischemia-reperfusion, very limited preservation times. And as Agnes was showing you with regard to the lung xenografts, their survival is in the period of hours to days. Hearts, much less impacted by ischemia than lungs, but obviously worse than livers, and then kidneys, as I say, you can keep them for 24, 36 hours. What's curious is cellular rejection. Again, it impacts hearts very, very dramatically, much more so than kidneys. And livers, usually, we get around the cellular rejection very promptly with immunosuppression. The humoral rejection, resistance to ABO barriers, again, hearts, kidneys, and livers in this order. We can transplant a liver across blood group barriers in an emergency. You're unable to do that in a heart. If you are transplanting across ABO barriers, you have to take then precautions to prevent elicited antibodies being generated. But certainly the liver is much more resistant to antibody-mediated rejection than you would get with a heart or a kidney. And I find this also very curious. With chronic vasculopathy, the chronic rejection, this is the major problem with transplants in the allotransplant situation. Highest rates are in cardiac transplants, about 50% within 5 years. Comparable numbers in kidneys, but it takes twice that duration to get that level of chronic rejection. And in livers, we're only seeing it about 10 to 30% at about 10 to 15 years. So much less in the way of chronic rejection. And again, much less in the way of humoral rejection with regard to the livers. But I think the main mechanism in the liver, which is different, is that your donor venous endothelium is replaced by those of the recipient. So if you, for example, these are images from a paper in the Lancet. If you take a graft from a female liver, transplant it into a man, then you look at different time points, you'll see that there is replacement of the venous endothelium by basically recipient cells which have come from the bone marrow, formed new endothelial cells in the actual microvasculature. And that clearly then will allow the graft to act as a scaffold and for the endothelium to become those of the recipient, which will not be then subject to any alloimmune stress. That does not happen in the heart, for example. It's a unique property of the liver. And this may be why also the liver has this unique capacity to regenerate. You can take out half of the liver, transplant it, and the actual donor who's lost half the liver will grow the remainder of the liver within a period of several months. And that you can keep on doing that in animal models. You can keep on doing rat liver regeneration experiments for the duration of the lifespan of the rat. The liver just has this unique capacity to keep repopulating itself. And it does it at least in part from recruiting bone marrow-derived cells. So why is this heterogeneity? Why is it present? Does it have any relevance for the heparan sulfates? If you're looking at the hemangioblast, the angioblast, there are these high notch pathways which lead to arterial fate, and then this low notch induction which leads to a default path with a venous fate. But it's very complex. This work has been developed by Bill Erd after initiation by Bob Rosenberg, who Corby worked with as well, MIT. And essentially what you're looking at, the microenvironmental effect going further as you go from the hemangioblast, progenitor cells, to artery, vein, capillary, and then these unique microvascular heterogeneity very well dictated by the microenvironment, by growth factors, possibly by matrix, by heparan sulfates, GAGs, other factors in the environment, whereas the epigenetics much more important in the earlier pathways and not so relevant to the heterogeneity of the vasculature. And again, with regard to hemostasis, if you're looking at endothelial cells from the liver, remember this is the endothelium which is fenestrated. It has big gaps in it. It allows platelets to move into the actual sinusoids from the actual blood flow. It allows chylomicrons to circulate. If you look at the breakdown of the procoagulants and so forth, the amount of von Willebrand factor is there, the amount of PI1, plasminogen activated inhibitor, is very high, and it's counterbalanced by high levels of TFPI, thrombomodulin, where when you look in the lung, high levels of heparan sulfate, much greater than in the actual liver, but note also the high levels of TPA relative to the liver. This lung pattern is also mimicked by what you see in the heart. And clearly when you start using drugs to treat myocardial infarction, heart attacks, one uses high levels of fibrinolytics, which are very successful at reopening the arterial flow, not only in the lung, but clearly in the heart as well. So this hemostatic balance, not only is it different from organs, but it's also different to the level of the microvasculature you're in. These endothelial cell genes, von Willebrand factor, CD39, this ectoenzyme of our interest that I'll talk about later, high levels in veins, arteries, then hepatic endothelial cells, and then high sinusoidal endothelium, brain and lung arterioles with TPA, these protein C receptors, large veins and arteries, TFPI only in the microvasculature. And curiously, tissue factor that we hear so much about initiating clotting is not present in the quiescent endothelium. It's just not there. You have to get endothelial cell activation to actually drive a clot. So the endothelium normally is anticoagulant, right? You're not getting a blood clot. To start initiating clotting, you have to start inducing the tissue factor. And you do that with inflammation, with TNF, IL-1, IL-6, other modalities like that, or antibody binding, complement-mediated activation. Does it work? Yeah. So let's talk about vascular bed specific inflammation and how is it relevant to what the focus of my own laboratory is. Maybe there are some overlaps with the GAGs and heparan sulfates, but we've been interested in this purinergic signaling pathways. So these are purines, they're nucleotides and nucleosides. It's been recognized from 1929 onwards. If you injected ATP or adenosine, you had major cardiovascular effects. And if you're in cardiac practice, you inject adenosine to actually slow heart rates down in people who are very subject to tachyarrhythmias, to arrhythmias. It works very, very quickly. Slows the heart rate down and then puts them back into a normal rhythm. ATP itself can cause bronchospasm, but it also has cardiovascular effects. So it's clear that these are not just working as energy mediators inside the cell, that they have functions outside the cell. But everybody poo-pooed it. They said, oh, well, this is probably just artifact, or maybe just effect of some other contaminants, that maybe the phosphate that you're injecting. Then Jeff Bernstock, who was an Australian boxer at university, got into the field, and he started looking at synaptic neurotransmission. And again, it was good that he was a boxer, because everybody poo-pooed him. They said, no, what you're looking at is rubbish. It's all artifact. You're just stretching the nerves, and that's the injury model. But he continued with this work. And it was good that he actually started documenting nucleotide receptors. A whole series of these G-protein coupled receptors were cloned. In addition, they were able to document how nucleotides were released from cells. And then you started developing drugs like clopidogrel, which is an ADP receptor antagonist, which is probably the second highest grossing drug now sold in the market. The highest used to be these statins. But clopidogrel is the same as almost like a country's GDP. In fact, the market for clopidogrel is greater than the bottom third of all the countries in the United Nations. So it's about $10 billion a year just on selling a drug to target platelet receptor. Clearly, it's very effective at cardiovascular disease and stroke and myocardial infarction with stents. And our contribution has been looking at vascular and immune cell activation over the last 10, 15 years. And our interest has been in looking at these vascular ectonucleotidases. So, to quote Einstein, make everything simple as possible, but not simpler. And UTP, ATP, nucleotides, how are they released from cells? They're in platelet granules. So the granules on the platelet, the dense granules, have molar concentrations of nucleotide. It's packed like a factory shelf with just nucleotides, no water, just basically anhydrous nucleotides. And when platelets are activated, they release very high concentrations of the nucleotide, millimolar concentrations at points of thrombosis. And this obviously can activate receptors. But under basal conditions, it can be released by connexins and hemichannels, and there may be also an ATP transporter to allow ATP to be released from a cell to sort of auto-regulate its cell size. With inflammation, clearly you get increased ATP levels. You can actually measure it by luciferase using the same mechanism of biophotons that the firefly does. So you can inject a luciferase construct, do the experiments to look with graft versus host disease, where you're putting in bone marrow which is foreign to the host, the bone marrow, and almost a reversal of a reaction, a rejection reaction, starts responding against host cells. And you can see where you start getting intestinal GVHD, that you're lighting up with photons with the luciferase, suggesting that ATP is being released. So ATP is released from granules, from cells, from channels, present at sites of inflammation, as in this paper. What are the receptors? They come in two main flavors. So there's the P2X, which is a ligand-gated channel, which there are 7. They only recognize ATP. They don't recognize ADP or UDP, just ATP specific. And they form a channel. They're important in calcium flux, potassium flux. Then there are these G protein coupled receptors, of which there are about 7 or 8. Some of them were thought to be UTP or ATP receptors, but later was shown that the actual affinities were not that great. So you look at the literature, you'll see P2Y1, Y2, oops, no Y3. It was sort of discredited. Y4, Y6, and so forth. And those are a little bit more non-specific. They'll bind UTP, UDP, ATP. And the platelets, for example, will have 3 receptors, all for ADP. So it'll be P2Y1, Y12, and X1. And the reason that cells accumulate lots of receptors is because ATP is fairly ubiquitous. So if you want to get a specific response, you control the release of the ATP, but then you put on different receptors on the cell which make it fine-tune it. So it'll only respond to a different concentration of ATP, for example, high versus low, or a different UDP after it's lost a phosphate. And the way it can lose phosphates is by the presence of ectonucleotidases. But again, this is other work with P2X7 and GVHD. I'm showing that if you do this irradiation model and bone marrow transplantation, then you actually start looking at a positive feedback loop. You're dependent on the presence of P2X7 on the bone marrow-derived cells to initiate injury in graft versus host disease. Again, that was similar work to what I showed you with the ATP lighting up in the small bowel and the point of the GVHD. So P2X7, very important receptor of any of these. It's called a death receptor. It triggers macrophage activation. It's called basically a very important mediator in regard to inflammatory bowel disease, transplant rejection, and GVHD. So nucleotides seem to be pro-inflammatory. If you look at the actual presence of CD39, it couples with an ectoenzyme called CD73 to generate ultimately adenosine. So you have ATP, ADP, CD39 takes them both to the monophosphate, CD73 generates adenosine. Simplistically, nucleotides tend to drive inflammation. The ectonucleotidases convert them to AMP and to adenosine, which are anti-inflammatory and tend to dampen inflammation, tend to promote healing, and may also promote fibrosis. The adenosine works by different adenosine receptors, which are all G protein coupled receptors. And they work, again, different levels of concentrations of the nucleosides. A2A will, for example, work on nanomolar concentrations of adenosine, whereas the A2B will work only on micromolar concentrations. So one is much more sensitive. This one's present on T cells, for example. This one's present, which responds to high concentrations on the endothelium. And again, they're all coupled to adenylate cyclase. They signal through cyclic AMP. So adenosine is an innate anti-inflammatory metabolite. It's widely expressed. It's also present in most of the preservation fluids that we use for transplantation. Just by trial and error, looking at different components in the 50s and 60s, the compound that actually works is adenosine in UW solution, University of Wisconsin solution. So we're using 5 millimolar of adenosine in these preservation fluids even before these receptors were characterized. And the A2A receptor is the most extensively characterized. So you can use these drugs. They're very important in protecting against ischemia-reperfusion injury. As I've said, they're present at high levels in preservation fluids. In the liver, this is initiated by NKT cells. Just suffice to say, the pharmacological effects of adenosine agonism are mediated by the A2A receptor, because in the A2A knockout mouse, you lose protection. In renal ischemia-reperfusion injury, again, you need A2A receptors, but not only on the endothelium, on the circulating cells, which mediates protection against the injury. But again, our interest has been in CD39. It's got two transmembrane regions, a very large ectodomain, which is heavily glycosylated and decorated with carbohydrates. I won't bore you with all of this data, but it's essentially a major mediator of adenosine generation. It blocks inflammation in experimental models of liver, kidney, bowel, cardiac, lung, brain, and islet, ischemia-reperfusion injury, stroke, and transplantation. We've generated transgenic mice. Keiichi, who spoke this morning, was the first to make the knockout mice in this family of molecules. And we've also, working with the Australian group, generated CD39 transgenic swine that David has been looking at. Keiichi showed you this work with regard to the thrombus formation in the microvasculature and the role of CD39. I just want to show you one further element of that. So Keiichi showed you the platelets and the fibrin labeling, and he also showed you this one, the CD39 in the green over here. This is the CD39 image. As the thrombus forms, do you remember when he showed you this morning, it didn't just keep on growing, right? It got to a certain size, then it started shrinking. And we think one of the mechanisms why it starts shrinking is it starts accumulating CD39 within the thrombus. So there's the hole in the endothelium where you've used the laser, and there's the actual thrombus. It's growing and it starts accumulating CD39, starts generating adenosine from inside, and then the clot itself starts basically retracting. You're no longer recruiting platelets, you're generating high levels of adenosine. Adenosine itself is antithrombotic, anti-aggregatory. And Keiichi's been looking at this further detail. It looks like it's bone marrow derived cells which are producing microparticles which then accumulate in the actual thrombus. So that's quite an interest. We've published on the microparticle component. I won't go into that in any more detail. But in the transgenic, what David was asking this morning, the CD39 does accumulate in the thrombus, and you can see in this composite figure. So that maybe is one mechanism how the thrombus basically doesn't keep on propagating and cause disseminated intravascular coagulation because of the accumulation of CD39 within the thrombus. So that's good, but the problem with CD39 is it's very subject to oxidative injury. And we published way back that if you actually were subject to ischemia-reperfusion injury, that you had loss of the NTPDA's biological activity. So the enzyme is dependent on its accumulation in lipid rafts. If there's any lipid peroxidation, you lose a multimeric structure of the enzyme, breaks down, and then you lose enzymatic activity. However, you're able to induce CD39 by transcriptional upregulation, which is brought about by hypoxic stress. So with hypoxia, HIF1 alpha and SP1 domains are important at upregulation of the gene. So it means that, in essence, you can precondition a graft. You can actually cut off blood flow for a few minutes, then allow reperfusion. Cut off blood flow for a few minutes, allow reperfusion, then do your harvesting. And this subjection of the graft to hypoxia is called preconditioning. And at least some of the actual protective mechanism is due to CD39 upregulation. So in the absence of CD39, I'll just show you a few examples. You do an ischemia-reperfusion injury, even in just a one gene deficient animal, not even a full knockout, you start getting this massive venous thrombosis, hemorrhagic necrosis, and vascular leak. And the survival curves for hemizygous and knockout are measured in minutes after ischemia-reperfusion of the liver. And you get similar patterns whether you're doing ischemia-reperfusion of the heart and other organs. CD39 transgenics, hearts, kidneys, and livers seen over here are protected from ischemia-reperfusion injury because you have a much higher level. You've got a pharmacological level of CD39. The oxidative stress will kill some activity, but you're left with a residual functional activity which is able to overcome the injury and the oxidative stress. And this is the preconditioning experiment. if you do the preconditioning and then you do the ischemia-reperfusion injury, you limit the amount of liver injury which is generated by the ischemia. So the CD39 generation of adenosine is protective. You can either protect the CD39 by preventing oxidative stress, or you can upregulate it by preconditioning, or you can do a transgenic. If you're looking at a xenotransplant, you can make it transgenic. You've got a very high level which allows the graft to be protected. So let's talk about pigs. So Churchill liked pigs because dogs look up to us, cats look down on us, and pigs treat us like equal. And what we're trying to do with these pigs is make them more equal by putting in human genes like CD39 and other thromboregulatory factors. And there's a reason to do that, and I'll show you what the reason is. See, some of these pig thromboregulatory factors don't work well across species barriers. You're putting in a pig graft into a baboon or ultimately a man, and some of those anticoagulants which are expressed on the endothelium don't work well across species barriers. So if you're putting in human genes, it's a way around that. So the only reason we're doing this, I mean, allotransplantation is fine, but there are very few donors, which is good because the roads are somewhat safer. You know, we used to kill 50,000 people every year on the roads in America, and now it's like 32,000, 33,000 because of better, drunk driving rules. Kids are better trained, and seatbelts in cars are safer. That's good, but there's still a shortage of donors. People die on waiting lists. They're on waiting lists for 1 or 2 years, and they die. So if there was a way to give them a graft off the shelf from a primate or a pig, obviously that could be life-saving. We don't like doing grafts from monkeys and old world monkeys or apes. They're concordant. They don't have acute rejection because they have the same alpha-gal patterns that we have. New World monkeys that David pointed out, when did you do that? About 10 years ago? They're different, aren't they?
They express gal, but they're so small.
Yeah, and they're small. Yeah, but so the South American monkeys are different to the old world monkeys and apes, which are Africa. We're out of Africa, so we obviously there was a different divergence of the primate groups. But these are sentient beings. They require socialization, and people don't like killing them just on for donors. Whereas pigs, unfortunately, 600 million pigs a year, is it? Something like that in America. The figures of chickens are even worse. I mean, chickens is what, 30 million chickens a day in America? But pigs are a lot, and it may not be 600 million. Isn't it 80 million or 100 million?
It's 100 million
pigs.
Pigs in America for food, but I was told today 600 million for heparin in China.
It's China, that's where I saw that data, yeah. So it's 80 to 100 million for food in America. Yeah. So basically pigs get sacrificed, for the common good. So if these animals could donate organs, if they were kept in certain circumstances and they were genetically modified, it might work. The problem is it's a discordant combination. So Roy Kahn way back showed that if you have a concordant graft, it doesn't get hyperacutely rejected. So hyperacute rejection, it's gone within 24 hours. So you put it in at night, the morning it's gone. Not much good clinically, really. So a discordant species is hyperacutely rejected. Concordant species, there's no hyperacute rejection. But the good news about the pig is that 90% of the antibodies which cause the hyperacute rejection are to this epitope. And you can get rid of that by doing the knockout for the enzyme which generates that. This figure was shown to you earlier. Essentially, we've been working on this I've been working on it 15 years, when we looked at antibodies complement overwhelming the heparan sulfate, the thrombomodulin, TFPI, inflammation, and clotting, which is a major component. In the hyperacute rejection, you get clotting and hemorrhage. In the delayed xenograft rejection, when you got rid of complement, you still ended up with the fibrin and the platelets. So you get around that by taking the gal out, so you lose the actual xenoreactive natural antibodies, so the graft survives longer. But the problem is we've been thinking recently that the gal deletion, you'll win in the short term, but maybe long term, you may also provoke ongoing vascular injury because potentially that gal deletion may make the gags and the vasculature more prothrombogenic. So David was asking about tissue factor, the role of tissue factor pathway inhibitor in this context. This is the actual model tissue factor by generating 10A, prothrombin. This is TFPI, 10, 7A tissue factor. The 10A is neutralized by the Kunitz 2 domain, and simplistically the tissue factor 7A interacting with the Kunitz 1 domain. We were able to show that this Kunitz 2 domain was different sequences, different properties in the pig, and the pig endothelial cells were basically inducing human tissue factor and failing to inhibit 10A. And again, this was work for over a decade ago, that there was an incompatibility of the TFPI with regard to the inability to neutralize primate or human 10A. Note also that this tissue factor pathway inhibitor is bound to a GPI-linked co-receptor but associated with GAGs. So when you lose the GAGs on the endothelium, when it's activated, the TFPI is also shed. Antithrombins in plasma and associated with GAGs. When we did the initial studies on this, we found that, like porcine intestinal heparin, some of these GAGs were actually fairly effective at blocking human and primate coagulation. So we did not find much of an incompatibility with wild-type antithrombin and GAGs with regard to the pig-human interaction. But we did see a problem with thrombomodulin, which is a problem at the top of the cascade and a problem at the bottom. And thrombomodulin, as you realize, is altering catalytic specificity of thrombin by binding it. The thrombin is no longer operating now on fibrinogen to form fibrin. It starts operating on protein C, which is bound by the endothelial cell protein C receptor to generate activated protein C, which has a negative feedback on factors 5 and 8 in the coagulation cascade. But again, pig thrombomodulin was incompatible with human thrombin, as shown in these papers with the autocatalytic activation of human prothrombin by endothelial cells from the pig, by Jeff Platt, who had also done a lot of work in heparan sulfates in the xenotransplantation context, and the more recent paper by us with correcting this by upregulating human thrombomodulin cofactors in pig cells and correcting the xenotransplantation incompatibility. We also published on the pig von Willebrand factor. It was known when hemophiliacs received factor 8 concentrates from pigs that it led to thrombocytopenia, and this was due to contaminating von Willebrand factor, which interacts with primate and human platelets in the absence of shear through a modification of the GP1B interaction on platelets. So in the absence of anything else, absence of any clotting problem, the platelets themselves from a primate will end up being very sticky with regard to the pig endothelial cell because of the von Willebrand factor. So the coagulation in xenografts, these are the key players: antithrombin, GAGs, TFPI, CD39 that I alluded to, thrombomodulin, the endothelial cell protein C receptor. So this is the pig graft sitting happily in the pig. You do the transplant, ischemia-reperfusion injury, you start generating lots of ischemic stress, reactive oxygen species, ADP, and so forth. The endothelial cells retract. You expose the endothelial matrix and von Willebrand factor. CD39 and thrombomodulin are inactivated by the oxidative stress, we published. And then you get the GAGs shedding. The CD73 is also shed through the GPI anchor, which is subject to phospholipase C activation. And so you've lost all these anticoagulants. And you start generating then tissue factor. So you lose anticoagulants, you start generating procoagulants. There's the tissue factor. You also generate a unique factor on the pig endothelium, FGL2, which is also important in generating thrombin. So you get beyond that. You've removed some of the actual antigens, you've removed the xenoreactive antibodies. The graft recovers, but it's subject then to quasi-recovery because it's only partially recovered because of these incompatibilities. So these pig anticoagulants don't work that well across species. And then post-ischemia, you start getting pre-rejection. You have a relative deficiency of APC. The levels of CD39 are low functionally, and TFPI is not operative. And then you start getting antibodies being generated against non-GAL, and you end up with the same situation again. All of these are shed, and the graft is eventually lost with vascular injury. Now, if you could add to this in the GAL knockout the fact that the subendothelial matrix may be now prothrombotic because of maybe some heightened chondroitin, maybe some other factors relating to the GAL knockout, that would obviously not be a good thing. You've got over the acute stress, the graft has survived, but there's an intrinsic flaw which is leading to this problem perpetuating. In kidneys, when David was in Boston, we were getting about 75 days, 90 days peak survival. So if you can put a kidney into a baboon, these are basically transplants where you've taken out the kidneys or ligated the ureters, so it's a life-supporting kidney. If you can get that through about 6 months, then you're looking at something that could be attempted clinically. But we're still not at that point. If you start using adenosine, you can actually improve things, but it's still not at the point where we could actually start looking at clinical trials. So what we thought about using was transgenes to upregulate the human factors. And Agnes and David have alluded to this. So the initial work was on regulating complement with CD59, 46, and DAF. I mentioned that the liver was very resistant to antibody-mediated rejection, and that's possibly in part because the liver has very high levels of these on the vasculature to start with. But this was the initial transgenic pigs. Then the knockout of the galactosyl transferase, a GAL knockout, which was a major quantum jump. Then our approach has been with these anticoagulants, with Revivicor and with the Australian group, looking at thrombomodulin or tissue factor pathway inhibitor and CD39. So the CD39 transgenic pigs, there's no overt bleeding. Agnes and Robin have looked at them in Maryland studies. David has done some transplants of the heart with these, and we've done some work in cardiac ischemia-reperfusion injury and renal transplants. We've also got another approach, because we don't want to just put in 1 or 2 transgenes. We maybe want to put in up to 6. And then Peter Cohen developed this F2A approach, where you make a single construct with these different open reading frames modified on hand, foot, mouth virus. The mRNA is a single message, which is then translated. You get the 2A processing, and you get the different proteins made at equimolar concentrations. So we've done this with CD39 and thrombomodulin on the background of GAL knockout and complement regulators with the H transferase. And we've had 9 liters of these, which have been used in transplant studies, in cardiac transplants, ischemia-reperfusion injury, and islet transplants, which is ongoing at Emory. And a couple of papers. One is just the cardiac injury with ischemia-reperfusion and ligation. The infarct size in a wild type is markedly reduced by the ability to generate adenosine and have high levels of CD39. So this is not a transplant model, this is a myocardial infarction model where the actual transgene has been very protective at adenosine generation. Just point you, I'm not going to spend time talking about CD39 on T regulatory cells, on dendritic cells. I'd refer you to this New England paper. We did a recent review on this. It does summarize some of the effects of ATP and ADP on the vasculature, how it's important in neutrophil and other inflammatory cell recruitment, and we go into the mechanisms of platelet activation in some detail. So I'll just refer you to that. So in summary, I've shown you about endothelial cell functions on coagulation, how it's important not only just in ischemic stress, but also in rejection and chronic rejection, and curiously also in bone marrow transplantation, where you're not really transplanting vascular substances, but those cells are able to come along and attack the endothelial cells. We think adenosine generated by hydrolysis of ATP protects against ischemia-reperfusion injury, also has salutary effects on cellular rejection by working via regulatory T cells. So suppressive regulatory T cells also generate high levels of adenosine. And genetic or other modification of donor organs will bolster CD39 and these thromboregulatory factors, and we can afford vascular protection and clinically relevant transplant models in the mouse and increasingly in the pig models. I'm grateful to David for all the work, and this is the group from Boston. This is Keiichi and Joichi, a little bit younger, but he's there. And then these two developed a lot of the immunological parameters, regulatory T cells and CD39. And David's a nephrologist who's been working in inflammatory bowel disease. So I think I'll stop there. Thanks, David. Thanks, Agnes. Thanks, Robin. Thank you, Umesh, for the invitation. So questions? Come on, April.
So during transplantation, you basically remove all processes in terms of fluids and blood and oxygen. Why is it so necessary?
So you have to basically, you have to perfuse the organ to preserve it. So you flush out the blood to prevent any intrinsic clotting occurring during that time, and you replace it with preservation fluid, which is a sort of a balanced salt solution with hydroxyethyl starch, which maintains oncotic pressure and prevents cell swelling and leakage. And then adenosine is at present at high levels. The properties of these preservation fluids are different for different organs. For cardiac, it may be different to the liver. I'm a hepatologist, so we work with UW solution. That has to be done during the harvesting process. If you just took a graft out, then transferred it with just warm ischemia, you may get away with it. But having cold preservation, you do need the blood removed, otherwise it'll just clot.
The alternative would be to fuse it with warm blood the whole time.
Yeah.
Yeah.
They're transporting it across the country, that is.
That's
So shouldn't small motors be created to be able to do that?
That's done for kidneys. You can do it for kidneys. You keep them on a pump, but not done on the liver. It's not standard practice yet, but it may come about. I mean, the other alternative is if you could able to take the graft and immediately implant it, in the presence of antithrombotics or synthetic heparan sulfates. That might be a way around it.
But even when, for example, the transplant of the heart, even if it's the donors in the next operating room, we cool it down because it takes us 30 minutes to put the heart in. So during that period of time, when we can't be perfusing it with blood, away all the sutures and everything, so we still cool it down, protect it for that 30 minutes, like putting it in the fridge for 30 minutes while we stitch it in, then it's reperfused with blood. And even that 30 minutes, you can get quite a lot of damage.
And arrhythmias.
If you had left it at normal temperature.
Yeah, the other thing that's been done now is people, we used to use cadaveric organ donors, so the heart's still beating, but people are brain dead. So because of the shortage of donors, they've extended donor criteria. So now you transplant people when the heart has stopped, and they've been heparinized and then put onto cardiopulmonary, bypass, or the graft has been perfused locally, then that graft can be used. But again, there's much more complications because of the ischemia. So you want good.
Flash freeze it.
Just die. It's very complicated vascular structures. You could flash freeze skin.
Cells.
Yeah. Skin you could do. That's been cryopreserved. But then.
Let me ask you a question.
Yeah, David.
How many of these different transgenes are we going to need to solve all the problems without help from Kubi and Umesh with all their drugs?
We need help from Umesh and Kubi, and from the other group with the
Which ones are these
chemokines. So I think complement regulation is important.
I was thinking more with thromboregulation.
Okay, so complement regulation is part of that as well. Because complement will drive platelets and it'll drive endothelial cell activation. I think the GAL knockout has been an important advance. Depending on what we find in this current project with Kubi and Umesh, if we're finding that there's high levels of chondroitins and we sort of make that analogous step, that high induction of chondroitin is leading to some potential contamination of the graft, which is leading to thrombosis. There may be other ways that we can talk about, maybe bringing that down, suppressing that. Then thromboregulatory, I think tissue factor pathway inhibitor will be useful if we're able to get good level expression. We haven't been able to do that yet. You, I think, have got a better option.
It's not as good as we'd like.
Yeah, so TFPI is a difficult one. I'm not sure why. Whether it's the glycocalyx association. Thrombomodulin works better at regulating. You've got good upregulation of thrombomodulin in your studies as well with revivicor. So something at the top and the bottom of the coagulation cascade, TFPI, thrombomodulin. I still like CD39 as a thromboregulator for platelets, because even though you're not affecting that von Willebrand factor, you may be preventing platelets from activating by removing nucleotide. You could take away the von Willebrand factor, but then you've got von Willebrand diseased pigs, and then they tend to bleed a lot and have problems giving birth. And then, maybe some cell protective factors, hemoxygenase, A20, to prevent apoptosis. And I think then you could probably look at something which could serve as a bridge for 6 to 12 months, for a xenotransplant.
Do we need CD73 as well as CD39?
Probably not. I don't think you do. I think the CD73 is fairly ubiquitous. So the rate limiting enzyme is CD39.
But CD73 is shared with,
Yeah.
shown with phospholipase, say, about
Yeah.
activation, yeah.
What I'm relying on is just the facts that we've got in myocardial infarction with CD39 alone is very, very protective. But you're right. I mean, you could argue that CD73, if you make it transgenic in addition, you may end up with a problem because the high level of adenosine, it's very helpful in the acute setting, but over a protracted period, adenosine can actually lead to increased healing, so it can cause scarring. So drugs like methotrexate, which are used in rheumatological diseases, can cause liver scarring, lung scarring, thought to be due to adenosine. You know, people who drink Irish coffees are somewhat protected from alcohol. You drink 4 cups of coffee a day. You know, the caffeine and adenosine are antagonistic. So caffeine works by blocking adenosine effects, as well as the phosphodiesterase effect. So alcohol, some of the effects of alcohol and fibrosis are due to adenosine release. So people who are drinking 3 to 4 cups of coffee a day, their incidence of really severe liver disease is halved. Hospitalizations are halved. So the Irish coffee is not a bad idea. If you have to have 4 or 5 drinks of alcohol a day, take it with coffee. But CD73, if it's a high level of adenosine, you might get excessive scarring in the graft, or potential steatosis as well. I still think CD39 alone probably would be enough. But it's in microparticles. So it's from the membrane blebbing, it'll be microparticles. But it's not cleaved from the surface like CD73. Yeah, could be.
You obviously missed yesterday my presentation about angiogenesis and oceanic acid biology.
Yeah, sorry.
And so there I discussed about oceanic acid in particular human, and we have exclusively N-acetyl-sialic acid, neuronic acid, whereas pigs, and they have least amount of sialic acid. That's one issue. Humans, vasculature, we have more sialic acid than the pig vasculature and the pig organs. 3-4 more differences. And sialic acid now play a major role in immune regulations, innate immunity, and so forth. Another one is that not only levels of sialic acid, much less in pig organs compared to human organs and human vasculature, and also like you have N-glycosyl sialic acid that you have in pigs, but we don't have. Of course, we do produce N-glycosyl sialic acid containing glycosciences in cancer, but there is no clear, we don't have enzymes, genes to produce enzymes that make N-glycosyl, but we still incorporate that. Not gene work and others have claimed that since we drink milk and eat meat, and from that you get N-glycosyl sialic acid that somehow gets incorporated, that may eventually lead to inflammation and hence into cancer. And so when they have N-glycosyl sialic acid produced by this individual transmission, only endothelial cells will eventually replace the human endothelial cells eventually. But in the individual organs, tissues, the cells are still going to be foreign origin. They're going to still expressing N-glycosyl, and I don't know how much of that.
I think that's an important point that you raised with the sialic acid. I mean, obviously it's relevant in the flu season now, with the swine flu and the neuramidases and the sialic acid receptors, that pigs share both the receptors, shared by birds and humans, right? So they're able to act as an incubator and get the flu viruses recombined, so they'll affect from birds to pigs, then obviously from pigs to humans. If I remember correctly, though, I mean, the sialic acid differentiation in the pig is higher, correct? The levels?
The pig, much less.
Much less, sorry, it's much less. So that's right. So the acylic glycoprotein receptor, on liver cells, for example, might be a mechanism how we get thrombocytopenia with liver transplants that I didn't address here. It's less correct. So what happens when you do the transplant, you may be picking up preferentially platelets because it's recognizing a different level of sialic acid on the platelets.
The other thing about what you said, the glycosyl-uramic acid is only is present in all species except humans, even baboons and chimpanzees.
That's true.
So in our studies in non-human primates, we don't think it's playing a role from that point of view. But humans, as you know, have antibodies against it. So when we come to put this in humans, we might have to have a
That might be the problem.
knockout.
Yeah. So that was the other thing we were talking about. We hadn't really addressed, because that would only be relevant, I think, with liver, where you put in a liver pig graft, platelet counts plummet down, and that may be at least in part mediated by the acylic glycoprotein receptor on the endothelium on platelets, on the endothelium binding to platelets and causing their sequestration.
Don't you think your double knockout pig is health-wise much weaker than wild type?
A double knockout or a single? It's only one knockout so far, a gal knockout. But there are other there's a human transgenes. We're not knocking out the pig endogenous, although it would be a mechanism potentially to do it. But we're putting in additional transgenes.
But isn't it,
I mean, that,
The knockout, the same argument was said years ago when we were trying to knock out the gal, because people would say, well, maybe the gal is doing something important. If you knock it out, the pig's not going to be viable. And it was pointed out that we don't have gal, and we seem to be viable. And so I suspect if you knock out the new GC as well, it will still be viable.
Yeah.
The pig will still be.
but it may be a health issue. I mean, they may not breed as effectively. I mean, there are slightly different rate of cataracts in the gal knockout in the mice.
Only in that one clone.
Yeah.
That pig, not in the other clone.
Yeah.
Cloning phenomenon.
Yeah, but you're right. I mean, when you start playing genetics, with modification. But I mean, this seems to be going mainstream now. I read in The Economist and Scientific American about all of these polo ponies being cloned, and there are a couple of groups in Argentina just taking a couple of ponies and just cloning exactly the same. So instead of having all this different selection of ponies for polo, you have basically a whole stable of the similar horse, where you want exact standardization. I think it's still complicated. I think it's still breeding is probably a quicker way to get 80 to 100 million animals for food. But who knows?
okay,
Thank you. Thank you.